Hey there, Max Health enthusiast, and welcome to another learning episode. So today we're going to be learning all about glycolysis, and this is a very important topic because it helps you understand the whole metabolic pathway about the energy source of most organisms. Yeah. So let's get right into it. So what is glycolysis? It's actually a metabolic pathway and an anaerobic energy source that has evolved in nearly all types of organisms. And we actually have another name for it, for the process. We usually call it Emden Meyer health pathway. Emden. Depends on how you pronounce it. I'm not really good with this pronunciation. Yeah. And it's in honor of like the major contributors towards the discovery and the understanding of glycolysis. Although it doesn't require like oxygen, but its purpose in anaerobic respiration. Like it doesn't require oxygen. That's the reason why. That's the reason for its purpose in anaerobic respiration. Anaerobic, that means without oxygen, and it is also the first step in cellular respiration. So the process actually entails the oxidation of glucose molecules, the single most crucial organic form in plants, microbes, and animals. And most cells actually prefer glucose, although there are like a few exceptions, like maybe acetic acid bacteria that actually prefer ethanol. But in glycolysis, you have like two ATP molecules that are consumed and they produce four ATP, two NADH, and two pyruvate per glucose molecules. And the pyruvates can be used in the citric acid cycle or maybe serve as a precursor for other reactions. So, what are the fundamentals of this? So glycolysis ultimately it splits glucose into two pyruvate molecules. And you can think of glycolysis as having two phases that occur in the cytosol of the cell. So the first phase is usually called the investment phase, and this is due to its usage of two ATP molecules. Then the second phase is usually called the payoff phase, and these reactions are like they're all catalyzed by their own enzymes, like with phosphofructokinase being the most essential for the regulation as it controls the speed of glycolysis. Glycolysis can actually occur in both aerobic and anaerobic states. And in the aerobic states, in the aerobic conditions, pyruvate enters the citric acid cycle and then it undergoes oxidative phosphorylation and then leads to the net production of 32 ATP molecules. While in the anaerobic conditions, the pyruvate converts to lactate through anaerobic glycolysis. And anaerobic respiration results in the production of, you have two ATP molecules. Glucose is a hexose sugar, 6 sugar, meaning it is a monosaccharide with 6 carbon atoms and 6 oxygen atoms. So that's why it's hexose. Hexose stands for 6. So the first carbon has an attached aldehyde group, and then the other 5 carbons have one hydroxyl group each. So during glycolysis, like glucose ultimately breaks down into pyruvate and energy. Like a total of 2 ATP is derived in the process. So glucose plus 2 NAD plus 2 ADP plus 2 pyruvates plus 2 PI will give you 2 pyruvates plus 2 NADH plus 2 hydrogen plus 2 ATP and plus 2 water molecules. So the hydroxyl group allows for phosphorylation, the specific form of glucose that is used in glycolysis in glucose 6 phosphates. So for the cellular level, glycolysis actually occurs in the cytosol of the cell. And under aerobic conditions, pyruvate is derived from glucose. Pyruvate derived from glucose will actually enter the mitochondria to undergo like oxidative phosphorylation. Anaerobic conditions result in pyruvate staying in the cytoplasm and then to be converted to lactate by the enzyme lactate dehydrogenase. So for the molecular level, what happens is that glucose first converts the glucose 6-phosphate by hexokinase or glucokinase using ATP and a phosphate group. Glucokinase is a subtype of hexokinase, which is found in humans. Glucokinase also has a reduced affinity for glucose, and it is found only in the pancreas and the liver, whereas hexokinase is present in all cells. So glucose 6-phosphate is then converted to fructose 6-phosphate, which is an isomer, by phosphoglucoisomerase. I will show you I just want to say the explanation now. I'll show you the diagram later so you can actually have a picture of exactly how all this works. So the glucose 6-phosphate is now then converted to fructose 6-phosphate, which is an isomer, by phosphoglucoisomerase. And phosphofructose kinase then produces fructose 1,6-biphosphate using another ATP molecule. Dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphates are then created from fructose 1,6-biphosphates by fructose biphosphate aldolase, which is the enzyme. DHAP will now be converted to glyceraldehyde 3-phosphates by triose phosphate isomerase. That's the enzyme that's used to convert it, where now the two glyceride 3-phosphate molecules will now the glyceride 3 phosphate molecules will now continue down the same pathway. And glyceraldehyde 3-phosphates will become oxidized in an hexagonic reaction into 1,3-bisphosphoglycerate, reducing an NAD plus molecule to NADH and hydrogen plus. So 1,3-biphosphoglycerate will then turn into 3-phosphoglycerate with the help of phosphoglycerate kinase, along with the production of the first ATP molecule from glycolysis. 3-phosphoglycerates will then convert with the help of phosphoglycerate mutase into 2-phosphoglycerates with the release of one molecule of water. Enolase will make phosphoenolpyruvate, PEP, from 2-phosphoglycerates. Then, due to the unstable state of the phosphoenolpyruvate, PEP, pyruvate kinase will now facilitate its loss of a phosphate group so that it can create the second ATP in glycolysis. First, phosphoenolpyruvate will then undergo conversion to pyruvate. And what's the function of all of this? Why do we need it? Glycolysis occurs in the cytosol of the cell, just like what we said earlier. And it's actually a metabolic pathway that would create ATP, which is without the use of oxygen, but it can also occur in the presence of oxygen, so aerobic and anaerobic conditions. In cells that use aerobic respiration as the primary energy source, the pyruvates formed from the pathway can be used in citric acid cycle and go through oxidative phosphorylation to undergo oxidation into carbon dioxide and water. Even if cells primarily use oxidative phosphorylation, glycolysis can serve as an emergency backup for energy or as the preparation step for oxidative phosphorylation. So in highly oxidative tissue, maybe such as the heart, pyruvate production is actually essential for acetyl-CoA synthesis and L-malate synthesis. And this serves as a precursor to many molecules such as lactate, alanine, and probably oxaloacetate. Glycolysis actually precedes lactic acid fermentation, and pyruvate made in the former process serves as the prerequisite for the lactate made in the later process. So lactic acid fermentation is the primary source of ACP in animal tissues with low metabolic requirements and little to no mitochondria. So in erythrocytes, lactic acid fermentation is the sole source of, as they lack mitochondria and mature red blood cells have little demand for ATP. So another part of the body that actually relies entirely or maybe almost entirely on anaerobic glycolysis is the islands, so which is devoid of mitochondria. There's no mitochondria there, as their presence would lead to light scattering. So those skeletal muscles prefer to catalyze glucose into carbon dioxide and water during exercise, during heavy exercise, where oxygen is actually inadequate. The muscles simultaneously undergo anaerobic glycolysis and oxidative phosphorylation. So for regulation, glucose availability. So the amount of glucose available for the process regulates glycolysis, so which becomes available primarily in two ways. So regulation of glucose reuptake or regulation of the breakdown of glycogen. So glucose transporters actually transport glucose from the outside of the cell to the inside. And cells containing glucose transporters can increase the number of the glucose transporters in the cell's plasma membrane from the intracellular matrix. Therefore, they will now increase the uptake of the glucose and the supply of the glucose available for glycolysis. There are actually 5 types of glucose transporters. So you have the glucose transporters 1, which ones are present in the red blood cells, the blood-brain barrier, the blood-placental barrier. We have the glucose transporters 2, which are present in the liver, beta cells of the pancreas, the kidney, and the gastrointestinal tracts. We have the glucose transporters 3 that are present in the neurons. We have the glucose transporters 4 that are present in the adipocytes, in the heart, and the skeletal muscles. Then finally, we have the glucose transporters 5 that specifically transport fructose into the cells. And another form of regulation is the breakdown of glycogen. Cells can actually store extra glucose as glycogen when glucose levels are high in the cell plasma. And alternatively, when levels are actually low, glycogen can be converted back into glucose. So these two enzymes control the breakdown of glycogen. And glycogen phosphorylase and glycogen synthase. The enzymes can be regulated through feedback loops of glucose or glucose 1-phosphate or through allosteric regulation by metabolites or from maybe phosphorylation or dephosphorylation control. And allosteric regulators and oxygen, just like we talked about before, many enzymes are actually involved in glycolytic pathway. So by converting one intermediate to another, control of these enzymes such as maybe hexokinase, phosphofructokinase, glyceraldehyde, 3-phosphate dehydrogenase, and pyruvate kinase, they can actually regulate glycolysis. And the amount of oxygen that is available can also regulate glycolysis. So, I don't know if you've ever heard about the Pasteur effect. And this Pasteur effect describes how the availability of oxygen diminishes the effect of glycolysis. And described availability leads to the acceleration of glycolysis, at least initially. So the mechanism is responsible for this effect. The mechanism that's actually responsible for this effect is the allosteric regulators of glycolysis and the enzymes such as hexokinase. So the Pasteur effect appears to mostly occur in tissues with high mitochondrial capacity, such as myocytes or hepatocytes. And still, this effect is not even universal in oxidative tissues, like maybe the pancreatic cells. Yeah, if you look at enzyme induction, another mechanism for controlling glycolytic rates is transcriptional control of glycolytic enzymes. So altering the concentration of key enzymes and allowing the cells to change and adapt to alterations in hormonal states. So, for example, increasing glucose and insulin levels can increase hexokinase and pyruvate kinase activity, therefore increasing the production of pyruvate. Then, for PFK1, that's fructose 2,6 biphosphate, is also an allosteric regulator of PFK1. High levels of fructose 2,6 biphosphate increases the activity of PFK1. Its production actually occurs through the action of phosphofructokinase 2. PFK is phosphofructokinase. Yes, so phosphofructokinase 1, that's PFK1. And the production occurs through the action of phosphofructokinase 2. And PFK2 has both kinase and phosphorylase activity, and it can transform fructose 6 phosphates to fructose 2,6 biphosphates and vice versa. So insulin dephosphorylates PFK2, activates its kinase activity, which increases fructose 2,6 biphosphate and subsequently activates phosphofructokinase 1. Glucagon can also phosphorylate PFK2, which activates phosphatase, transforming fructose to 6-biphosphate back to fructose 6-phosphate. And this reaction decreases fructose 2,6-biphosphate levels and decreases PFK1 activity. And that's it for the regulation. For mechanism, that's for glycolysis phases. Glycolysis has two phases. So like we said earlier, the investment phase and the payoff phase. So the investment phase is where there is energy, like the ATP is put in, and then the payoff phase is where the payoff phase is where the net creation of ATP and NADH molecules actually occur. So the total of 2 ATP goes in the investment phase with the production of 4 ATP resulting in the payoff phase. So thus there is a net total of 2 ATP, and the steps by which ATP is created is usually called the substrate level phosphorylation. So what happens in the investment phase? So in this phase, there are 2 phosphates that are added in glucose. There are 2 phosphates added to glucose. Glycolysis begins with hexokinase phosphorylation, phosphorylating glucose into glucose 6-phosphate. This step is the first transfer of a phosphate group and where the consumption of the first ATP takes place. Also, it is an irreversible step. So this phosphorylation traps the glucose molecule in the cell because it cannot readily pass the cell membrane. So from there, the phosphoglucose isomerase, the enzyme, isomerizes glucose 6-phosphate into fructose 6-phosphate. Then phosphofructokinase, PFK1, adds the second phosphate, PFK1, uses this as the second phosphate, and then PFK1 uses the second ATP and phosphorylates the fructose 6-phosphate into fructose 1,6-biphosphate. This step is also irreversible and the rate limiting step. So in the step that follows, fructose 1,6-biphosphate undergoes lysis into 2 molecules which are substrate of fructose biphosphate aldolase to convert it into dihydroxyacetone phosphate, DHAP, and the glyceraldehyde 3-phosphate, G3P. So DHAP is now turned into G3P by triose phosphate isomerase. DHAP and G3P are in equilibrium with each other, meaning they transform back and forth. And that's it for the investment phase. So for the payoff phase, what happens is that it's critical to actually remember that there's a total of 2 3-carbon sugars for every 1 glucose at the beginning of this phase. So the enzyme glyceraldehyde 3-phosphate dehydrogenase metabolizes the G3P into 1,3-diphosphoglycerate by reducing NAD+ into NADH. Next, the 1,3-diphosphoglycerate loses a phosphate group through phosphoglycerate kinase to make 3-phosphoglycerate and creates an ATP through substrate level phosphorylation. So at this point, there are 2 ATP produced, one from each 3-carbon molecule. the 3-phosphoglycerate turns into 2-phosphoglycerates by phosphoglycerate mutase, and then enolase. And then enolase turns to 2-phosphoglycerates into phosphoenol pyruvate, PEP. And the final step, pyruvate kinase turns PEP into pyruvate and phosphorylates ADP into ATP through substrate level phosphorylation, thus creating 2 or more ATP. So this step is also irreversible. So overall, the input of one glucose molecule is 2 ATP, and the output is 4 ATP and 2 NADH, and then 2 pyruvate molecules. So in cells, NADH must recycle back to NAD+, so that it can allow glycolysis to keep running. And if there's an absence of NAD+, the payoff phase will come to a halt, to actually stop and then result in like backup in glycolysis. In aerobic cells where oxygen is used, NADH recycles back into NAD+ by way of oxidative phosphorylation. And also in anaerobic cells, this occurs through fermentation. And there are 2 types of fermentation, the lactic acid and alcohol fermentation. So to just briefly give you like a detailed explanation of what's going on in this diagram. So a phosphate group is actually transferred from ATP to glucose, making glucose 6-phosphate. So glucose 6-phosphate is more reactive than glucose, and then the addition of the phosphates also traps glucose inside the cell, since glucose with a phosphate can't readily cross the membrane. So here glucose 6-phosphate is now converted into its isomer fructose 6-phosphate. Then this next step here, a phosphate group is actually transferred from ATP to fructose 6-phosphate producing fructose 1,6 biphosphate. And this step is catalyzed by the enzyme phosphofructokinase, which can be regulated to speed up or slow down the glycolysis pathway. Then in this 4th step, fructose 1,6 biphosphate will split to form 2 3-carbon sugars. That's dihydroacetone phosphate and glyceraldehyde 3-phosphate. And they are isomers of each other, but only one, that's glyceraldehyde 3-phosphate, can directly continue through the next step of the glycolysis here. And then this 5th step, the dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. The 2 molecules exist in equilibrium, but the equilibrium is pulled like strongly downward. So in the scheme of the diagram, like above the diagram here, that's just like glyceraldehyde 3-phosphate is used up. Thus, all the dihydroacetone phosphate is eventually converted. And then here the 2 half reactions now occur simultaneously. The glyceraldehyde 3-phosphate, that's one of the 3-carbon sugars that is formed from this initial phase up here, this blue side, is now oxidized, and 2 NAD+ is reduced to NADH, and this 2 NAD+ is reduced to NADH and 2 hydrogen molecules. So the overall reaction is hexagonic, releasing energy that is then used to form phosphorylates the molecule, forming 1,3-biphosphoglycerate. Then the next step, 1,3-biphosphoglycerate donates one of its phosphate groups to ADP, making a molecule of ATP and turning into 3-phosphoglycerates in the process. Next, we have 3-phosphoglycerates, which is converted into its isomer, 2-phosphoglycerates. And next, 2-phosphoglycerate loses a molecule of water and becomes phosphoenol pyruvate, PEP. And PEP is an unstable molecule poised to lose its phosphate group in the final step of glycolysis. And then this phosphoenolpyruvate is it readily donates a group to ADP, making a second molecule of ATP. As it loses its phosphates, PEP is now converted to pyruvate, which is the end product of glycolysis, and that's what we see here. So finally, we'll talk about the clinical significance, like, of everything. And pyruvate kinase deficiency is an autosomal recessive mutation that causes hemolytic anemia. So there is an inability to form ATP and then causes cell damage. And cells can become swollen and are taken up by the spleen, causing splenomegaly. And the signs and symptoms just include you have jaundice, you have icterus, you have elevated bilirubin, and splenomegaly. You also have arsenic poisoning that also prevents ATP synthesis because arsenic takes the place of phosphates in the steps of glycolysis. So thank you so much for being a part of today's exploration into glycolysis and seeing its clinical significance to your health and why you need to know it and understand that this is important for some cell functions that happen in our body. If you're able to gain so much value from this video, please give a thumbs up, subscribe, share, like, comment. It goes a long way in helping me create this free content to you. So thank you so much for listening. Talk to you soon. Bye for now.